Okay, good morning, everyone. I think we should start now. Thank you so much for coming to the Future of Technology in Genomics. I think this is probably a first for Global Alliance, as we are going to be experimenting with remote meetings over Zoom, which is probably really is the future of technology in genomics. I'm sure it will go without a hitch at all. I'm Andy. My co-chair Moran is here at the front. We're just going to give a brief introduction into the session, and we're going to introduce three very exciting speakers as well. At the very end, there will be a Q&A session. We would like you to reserve your questions until the Q&A session, so then you can ask questions holistically of all talks, not just of individual talks. One area that we need to start looking at is the global perspective around healthcare, because that really is in our future. This is a fantastic article that was released this year. It's talking about the integrating genomics into healthcare, and here's a map of all the national initiatives that are currently running that we're currently aware of. And you can just see that there is a huge number of national initiatives appearing from all around the world at the moment. So key to solving the problem of genomics is to actually embrace this global perspective. Part of building that global perspective, though, is also to actually start to embrace federation. And really, I think we've seen over the past years or so, this change from federation is now an expectation of how we operate, not the exception. Projects such as Seneca, which is a project between primarily Canada, but also Europe and South Africa, really is trying to bring together a global perspective around delivery of healthcare and genomics, and really making federation a reality. Standards are an increasingly important part of making this happen. Without standards, we can't possibly interop. Hopefully this is going to move. But then there's another side to this as well. There is the idea of fair. What is fair? Obviously, everyone knows it's findable, accessible, interoperable, and reusable as well. Fair ensures that the major barriers to actually data reuse are removed, and so that you can actually interrupt. Standards such as the data use ontology, researcher IDs, and even if you think back as well into formats such as CRAM, actually allow this to happen. And this is basically the key of how we do interoperability, how we do this federated network. But then there's another side to this as well. There is the issue of distributed data processing as well. There are problems here. Where is my code? How am I going to execute my code? Where is my data? Please go and run it. Standards such as the TRS, the Tools Registry Service, the Workflow Execution Service, and the Data Repository Service can all help here to actually address these problems. Again, though, there needs to be standards around data processing as well. I can keep on saying the word standards because I think that this is actually the key behind a lot of the challenges that we're facing at the moment. But this is a huge challenge that we need to address. So, moving towards a global future. I believe fundamentally that by you being here in this room and at this conference, you're already engaging in that future. The technology and the attitudes have moved to expect this global outlook. Again, you're here, you embrace it. Standards developed within the Global Alliance, but also standards developed outside of Global Alliance will also help to drive that. And the continued engagement and pushing the boundaries of what is possible will actually help us achieve that future. So with that, we have three speakers today. We have Barend from Leiden, we have Patrick from Princess Maxima, and also Sumit from the Global Gene Corp. Both Barend and Sumit are remotely presenting, so Patrick is actually our own only physical presenter today. Like I said before, I am sure this is going to work absolutely fine. I am really looking forward to the talks, and I am sure that a number of the things that I've highlighted here will be echoed through these other presenters. So with that, I'll pass over to my co-chair.
So just a brief introduction. So Barend is over there. Barend is joining us. He's a faculty at the University of Leiden and also a director of the Dutch International Support and Coordinating Office. And he's going to talk. He's a real expert on FAIR principles and advocate for open science. He's going to talk about FAIR today. Thank you, Barend.
Thank you. I hope everyone can hear me well. I'm really sorry that I cannot be there because there should be a lot of old colleagues and friends there because I worked on genetics for a long time and in Elixir. But Marco is there. As you see on my first slide, we do this sort of together. I give the talk, the easy part, and then all the difficult questions have to wait till the end. So Marco will be stuck with all the difficult questions, but he will be able to answer all of them, I suppose. So let me quickly go through, because then we can probably save you some time for discussion. As you see on my first slide, we now talk about an internet for social machines. And the idea there is that we now have such large data sets, I don't have to explain that to people like you, that we need machines all the time to help us to discover patterns and so on, but we still have to interact with them very regularly, and that's what we call a social machine, an environment where the machine and people work very well together. It was already mentioned in the introduction that we now go to distributed learning, which also poses other problems. So that's why I now usually start my lectures with the end of data sharing as we know it, because if you talk data sharing, most people think, I send my data somewhere else, I lose control, all the safety. We are here in the Leiden University Medical Center, we are not even allowed to do that. So FAIR has very little to do with open per se. I now summarize it as you see in the middle of the screen. The machine knows what I mean. That's a nice mantra to keep in your head. So you have to formulate everything in such a way with rich metadata that machines can become, for now, our data slaves, until they take over at some point. We'll see. So the idea is here that you have the internet of fair data and services, or the internet for social machines, where in fact you have three major elements. This is a sort of persiflage on the hourglass model of the internet. The tools, the algorithms, the virtual machines are somewhere, the data are somewhere, and the compute. And of course, when you run a job, an algorithm on some data, the compute has to be there, so these have to find each other in a very dynamic way, sometimes on your smartphone. Very distributed. And so the whole distributed learning by virtual machines, Docker images, etc., goes over more and more complex graphs that ideally should not just be data lakes, but already prepared in FAIR data. I will come back to a new meaning of FAIR that the Americans came up with very soon. But the more these data are machine ready, and machine actionable, the better the machines can help us. And as we all know, most bioinformatics people, for example, now spend 60 to 80% of their time data munching before they can start anything. So we could save many, many FTEs for research, and we expect that if we spend 5% of all research funding on this, we would be easily doing this. That's 10 billion a year in Europe, and it would save a multitude of euros more. So if we want to do this, we have to formulate the entire internet as what we call an internet of fair digital objects. This is the first artist impression that comes from a European project report on a fair digital object. So it's a bit sequence with content. Then it has metadata. It has an operations associated with it, so the machine knows also what it can do. And it has a persistent identifier. Now, we have taken that in the last 5 months a few steps further, and we actually did a lot of work in GO-FAIR, but internationally. We went to the United States, to China, to everywhere, to find the equivalent almost of TCP/IP in the internet. If we all formulate our stuff like this, the machines will just understand it, and we will break through a lot of barriers. So we now have a meeting planned in a week from now. That's one of the reasons I couldn't come, traveling too much, in Paris, where we will bring about 40 people together that have reviewed this globally, including some of the people of CNRI that were in the basis of TCP/IP originally, to come to a consensus. And the consensus is essentially this one. So if we really tune it down to what I always say when the real computer scientists are in the room, this is how a stupid biologist like me can understand this. Now, whenever a machine encounters anything in the Internet of Fair Data and Services, it should be able to answer these three questions. What is this? And then question 2, what operations are possible, technically, with that type of digital object? And then, of course, in the metadata, part of the metadata should tell the machine what it is allowed to do. So it's the license. Now, each fair digital record or digital object is a bit stream and has a type, and it contains many others, potentially. So it's a container in container in container, which is from the linked data platform. So it's a recursive concept here that the fair digital record could be as small as a single identifier, but even a single globally unique, persistent, and resolvable identifier needs metadata. For example, I want to know what the resolving mechanism would be, but also from which ontology is this triple coming, for example, or is this a unified UMLS identifier, an NCBI identifier? Then I know as a machine what I can do with it. And then in the end, as you see on the right-hand side here, if the machine is able to understand the digital object, it knows what it is able to do with this type, and it is able to resolve what it is allowed to do, then of course a digital record, a digital identifier or a triple, can refer to anything in the physical world, like a butterfly in Naturalis, here, a biobank, a sample in a biobank, a workflow, a database, or a single triple. So from the smallest object to the largest object, we have to build this internet. And if you do proper RDF nanopublications, for example, this is already an example of where you reach 90% of what we need to do. So this fair digital object could contain many other fair digital objects. A triple already has 3 identifiers, and then you build it up like this. Very importantly, the metadata are separated from the data itself, which is not common practice in many cases. And why is that? First of all, performance-wise. Secondly, if I am a machine and I encounter a very interesting data set, before I press download, I better know this is a petabyte of data, so I don't want to download it. So that's just 2 reasons out of maybe 10 why the metadata should be separated, but have a very strong permalink to the digital object itself, saying that the metadata itself are, again, a fair digital object of a different type. So it has the type metadata. Now, coming back to this could be a single identifier, or in this case, a triple. It could be a nanopublication with a lot of metadata associated and pointing to a particular resource. It could be a workflow in itself. So the actual thing it represents is a workflow. It has rich metadata, machine readable, so you know with which data sets this workflow can be run. And then the metadata of the data allow you to know which data can be used. And for example, if you are from a company and the data cannot be used for commercial purposes, the machine will know. That's just another GUPRI, globally unique, persistent, and resolvable identifier in the metadata here that refers to a machine-readable license. So if you now think about how we use this for our research, I will quickly give you some examples there. We have already published years ago the concept of nanopublications, where you have a single assertion, or sometimes a small graph, making an assertion. And of course now each of these 3, the subject, the predicate, and the object, are already fair digital objects, and they have their own globally unique, persistent, resolvable identifier. I always use my old example from my biology days. Malaria is transmitted by mosquitoes. The whole container has its own unique identifier. The provenance, in this case a part of the metadata, is also a fair digital object and has its own identifier. So they are always permalink, but separate, and they can be treated separately. So if you go to the next step of reasoning now, of course, if you take this example of the nanopublication, malaria is transmitted by mosquitoes, but it could be anything. This gene has a transcription start site there and there. You see that this triple is found in Medline more than 8,000 times. It's always the same assertion, malaria is transmitted by mosquitoes. But each of those nanopublications, look in the B panel, has a different provenance, different PubMed ID, different authors, and so on. But in the end, you really don't want to, I'll get a phone call, sorry. So you don't want to store these 8,000 triples. You basically have one cardinal assertion, as we call it, which is a new type. So the machine knows this container, although it contains exactly the same identifiers, is now a cardinal assertion and not a single nanopublication. So now I expect different kinds of provenance. I can do different things with this digital object, than I could do with the triple. Now, if you take it one step further, we have created so-called knowledge, and this is just one example of how you can use fair data, where if you take all the cardinal assertions, maybe 10 to the 6th, that have been done about the subject malaria, and you cluster them, as you see here in the knowledge, you have everything we have ever said about that particular concept. The knowledge, again, is a fair digital object, a larger container. It has its own unique identifier and its own provenance. And the interesting thing is that if we made a calculation years ago, that probably if we would be able to capture everything ever said in life sciences, we would have 10 to the 14th nanopublications, many more triples, of course. Then if the average redundancy of the same assertion would be 1,000, we would be down to 10 to the 11th cardinal assertions. But we only have less than 1 million knowledge because that's the number of concepts we publish about in the entire Medline database. So if you express those as vectors, you can do a lot of things. Now, don't think I'm just talking future or whatever. There's actually now a full company based on this called Euritos, and they have this knowledge as one of their basic ideas. And here you see 3 real knowledge, in this case of metabolic syndrome, diabetes type 2, and early onset Alzheimer. Maybe some of you know there seems to be a correlation that people that have these other syndromes, they have a higher chance to get early onset Alzheimer. And the machine tells you over, as you can see here, more than 3,000 resources, because for each of these triples, we have the full provenance, time, date, stamp, where they come from, etc., etc. And it shows you that only 5 proteins are shared in the middle, in the red circle, between these syndromes. And this you can do with much more complex queries than you do here. And normally this would take you weeks or months, and now it is 1 second, literally. So now with the idea that you can have many databases, the knowledge can also be the fair digital object that now acts as metadata of a database. So you can, for example, cluster databases based on their semantic annotation, and workflows can find data and the other way around. Here's another example of our own use of this. In the top, you see a nano publication, still an artist impression, of course, from one of the many millions that we made for the Phantom 5 data in the mothership paper. And there you basically see a description that for a particular gene, there is a putative transcription start site in a particular area between these 2 nucleotide locations on chromosome 2, with the reference genome, of course. And then you have a totally independent one, the green one, that is from the Leiden Open Variation Database, where doctors report deletions or SNPs or anything they find in the genome of people, and they probably relate them to a particular disease. Now, of course, as you can see, if you look at the numbers in the center, that the machine is now able to infer independently that this deletion is actually disrupting a putative transcription start site of that gene and not the gene itself. So now we can ask questions like, how many disease-causing variants, or at least shown to be potentially disease-causing by minimally 1 doctor, actually do not disrupt a gene but the transcription start site or any other of the regulatory sequences in the genome. And in this case, Rajaram, the guy you see here with this picture, came back via fair data within a minute with the answer that it was over 20%, which is an interesting result, which we won't go into the details now, what we can do with it. But those kind of complicated questions that would normally take you a lot of time can now be answered very easily, completely over distributed data. So you don't need to bring these data in one big database, the original data. So what can we do with this kind of technology? First of all, as I already showed, you have this idea that for any combination of concepts, genes, diseases, drugs, and so on, you now have a measure of similarity, conceptual similarity. And you can use that to predict drugs for diseases. We already did that and published that. But you can also use this now because you also have knowledge of other digital objects. So you have knowledge of databases, of workflows, and so on. And you know their similarity and whether they may be combinable. Now, it also solves another very, very serious problem in computer-assisted science, that computers cannot deal very well with near sameness. The predicate is nearly the same as means nothing to a computer. But assume that these three knowledge here would be actin mouse, actin human, and actin rat. Then 90% of the triples in there would be identical, but still the chromosomal location 14 and 4 in the mouse and the human, and Homo sapiens versus Mus musculus would be different. So if you would say to the computer that you don't really care about species or chromosomal location, these knowledge would actually merge into one. So you actually have an identical concept for now, and later you can split them again over species. This also means that the knowledge of a gene will change over time. So if there is a new assertion about the gene, for example, an association with another gene or a disease, then the knowledge will change. The GUPRI, the identifier of that knowledge, will change. But as a fair digital object, it will be traceable over time, so you can always go back to an older version. So you have a sort of a blockchain, and now you can deal with semantic drift, and you can show that, for example, genes move consistently closer to a certain disease over time, as we say, until even people get it, that they probably should have a look at that gene for that disease. The most exciting thing at the moment that we're working on is on the so-called concept of quas, because we have all the provenance, time, date, stamp, source, and everything for each of these assertions in the knowledge, we can filter. So from our field, you could say, I'm looking, let's say, at water as a very generic concept. If we want to go across disciplines and the whole internet, we see the concept water in the thesaurus of the C data cloud, of many other UMLS and Uniprot and so on, maybe. Chemistry, we can collect them all. They all point at the concept water. But if you only keep the assertions from one ontology, you look at it qua vision of the C data cloud, and they look at seawater, of course. So this is how you can filter knowledge and say, I don't care about species or about chromosomal location. Yes, now I do. And then the knowledge move in this dynagraph. So this is different from a static knowledge graph, okay? Now, coming back to realizing this, basically every digital record or every digital object would now be formulated as a digital object with this schema. It has its own machine-readable metadata, its identifier, and it points to something else. And they are stored in fair data stations. Maybe you saw the movie of the personal health train that then moves around and learns over these distributed data in fair data stations. And again, they can either point to another digital object, like a triple or whatever, but you can now have the physical object, a human, a butterfly, a sample, a molecule, a tumor, that we are starting a digital oncology center right now, where we have a digital twin of every tumor, which is essentially everything you have ever been able to record in digital and machine-readable format about that tumor or about that person. And of course, you see now in the University of Wageningen on the right-hand side that you now have the digital twin of a cow, the digital twin of a human, and we are even now starting to use these digital twins as researchers. So they could create virtual machines. A human could put in a little app, please check my variants for new information every week or something and report to me if they became associated with the disease or whatever. So finally, and then I will have I think I have a few minutes left, two or so. What I now say, if you look at knowledge discovery, then you see that on the right-hand side, I call this the gas phase, all these concepts are floating in space like Brownian movements. And then our knowledge gets to the right. It's the blue arrow here. And this is where most people discover knowledge. When these things become close enough, I call it the liquid phase, that even people see the pattern and we can do something with it. And an ontology, as we know it, or a static knowledge graph, is just a reflection of what we already know. So knowledge discovery, which we are all after, is here. But in many cases, and we have shown this in a model, we just don't have enough intelligence for pattern recognition as humans to see these patterns in highly complex data. And this is why we need fair data. And this is why I would like to end with this summary, where basically you say fair means the machine knows what I mean, as I said in the beginning. And of course, we strive also in the European Open Science Cloud for data to be as open as possible and only as closed as necessary. But for example, here in our hospital, we cannot give our data away, but I'm allowed to receive a virtual machine and run it on my data and only give the results away without traceable re-identification. We added to that, therefore, that we do things as distributed as possible. And I have a good bottle of wine for those who can tell me one question that cannot be principally solved by distributed learning. Performance reasons don't count. And only bring data together if it's absolutely necessary for whatever reason. And then the last thing to end with is, of course, our American colleagues, although they were involved originally in FAIR here in Leiden, of course, they always come up with a different term for the same thing, that we have a tradition there for elevator and lift and everything. So now they say, oh, you know, there is an interim report to NIH, and there they say all data should be AI ready. And I like that because it gives an indication that FAIR is not a goal in itself, but of course we could not resist to put the F word in front of it and say, okay, now it's fully AI ready, which means FAIR again. So with that, I would like to leave it, and you can ask all the difficult questions later to Marco. Don't be shy, make them really difficult, because normally I get them, but now I am off the hook. So thank you very much for your time. And again, I would have loved to be there, but this is all I could do for now. Thank you.
Thank you,
If there's
Barend.
any immediate question, I can answer, but I think you rather wait until the end. Eh?
I think so. Yes, thank you, Barend, and thank you for the new acronym. I really like it. It's a good one. So with that, I'm going to invite Patrick Kemmeren, who is a principal investigator in the Princess Maxima Center, and he's going to talk about setting up large-scale compute systems for studying pediatric cancers.
Okay, thank you very much. I'm going to try and see if I can squeeze my laptop in. Push that away slightly. Yes, this is going to be difficult. Have you got enough space? I think it should be fine. Okay. Yeah, it's stable enough like this. Okay. Thank you very much. Thank you also for giving me the opportunity to speak about our center and the kind of challenges and solutions that we ran into when setting up a new infrastructure from scratch. And just to give you a little bit of an overview, so I'm part of the Princess Maxima Center for Pediatric Oncology, which is a new institute in the Netherlands where we're dedicated to curing children with cancer. And we're not only performing healthcare there, but also have a big research department, which I'm part of. And we've initiated the center, so there used to be 7 different centers in the Netherlands treating children with cancer, and now we have one single center in the Netherlands treating all children with cancer, so it's about 500 to 600 children a year that got diagnosed with cancer in the Netherlands. And basically we started up a whole new institute in 2016, so the last couple of 3 years we've been working on setting up this infrastructure from scratch, which has, well, both is a blessing and a curse, I would say, because you're not only setting up kind of advanced infrastructure, but also talking about digital workspaces and all these kind of things, which I will not talk about here today. So in terms of our mission, So like I've already indicated, we're dedicated to treating and curing children with cancer. The childhood cancer survival rate has dramatically increased over the last decade to around 80% today. But despite these advances, still about the death in children is still primarily caused by cancer if you don't count traffic accidents in Western countries. And the aim within the hospital is really to go for 100% cure, to really treat these children and cure them with the best quality of life afterwards, after the treatment. And one of the goals we set out to do this in 2016 is basically to set up a whole biobanking initiative where we basically collect all tumor material from these patients and systematically collect them and also perform whole genome, whole exome, and RNA-seq based analysis based on these patient materials. And this is also the use case that I want to use to kind of highlight the kind of solutions that we took in setting up the infrastructure needed to support this kind of analysis. Just to show you the kind of different steps that are involved. So there's a number of steps when taking the patient or actually the patient material through the different steps involved. So of course we start with patient registration within the clinical EHR systems. Then we have sample collection which will occur, which uses either blood during regular clinical procedures or tumor biopsies taken during the operation when surgeons basically take the biopsies from the patients. Then we have a number of wet lab processing steps that occur where we basically enrich the material and process the material so we can actually do the data analysis and also do the sequencing. And then we end up with the bioinformatics, which is basically the part that I'm also heading within this whole part of the biobanking. And all these different steps also require quite a bit of infrastructure to actually facilitate and really accommodate these kind of different steps. And what we're really proud of is actually by investing a lot of time, and I apologize for this slide, is mostly in Dutch because I just took the screenshot from one of the reports we get from our EHR system, is that we've been really working hard using this infrastructure, using the reporting tools that we have to get the inclusion rates of the patients up. So basically the most important figure I want you to focus on is the one with all the bars in orange and blue. And what this shows is basically that we kind of want to make sure that we reach all the patients that enter into the hospital. So we want to make sure that every single patient that comes into the hospital is asked, well actually the parents are asked whether they want to participate in research. And everything which is blue is not good because we don't reach the patients. Because we basically don't hand out studies, so the biobank study is one of them, but it could also be part of multiple clinical trials. And then there's, of course, a diagnosis attached to this patient, which could be multiple diagnoses. And there's, of course, material attached to it, so the biosources and biomaterials, which are the physical samples that we collect during these different procedures. And then there's all kinds of systems that we build up based on this setup, which really cover a lot of different things. So we have, on the top end, we've got the electronic health record system, which we have a certain application for. Then we have applications on the left bottom, which are dealing with the kind of molecular diagnostic information, so the sample information coming from diagnostic lab, which is then translated into the biobank information system. And at the bottom right, which I will talk about during the second part of my talk, is a kind of genomic infrastructure that we've built up in order to support the genomic analysis and really help us analyzing the kind of genomic data that's coming through this setup. And in the center, then we have what we call the data integration analytics platform, where we're basically combining this data at a certain aggregation level in order to be able to connect all of this data together. And then there's a number of very important principles that we set out with in the beginning to really make sure that the platform works with all the different systems. So first, we have unique identifiers, which are institute-wide unique identifiers for both patients as well as samples. So all the resources, data resources that are located here should be aware of the kind of unique identifiers that we have defined, and we have a coding system for this, which makes sure that we have unique identifiers across all these systems. The tooling should be compatible with FAIR principles. Well, I won't talk about that because Barend already gave a very good introduction about the whole FAIR subject. And the other thing is by doing it in a FAIR-like fashion, we also encourage a modular setup. So we have defined REST APIs, FAIR data models, and that should allow us to plug and play, have like a plug and play architecture, allowing us to add new systems in the future as long as they comply with this kind of standards. So that's why I'm really happy with all the work that's been done also in GA4GH. And by doing that, then hopefully we can also automate quite a lot of the different steps involved in these different toolings that we have here. If we then look at it in terms of the kind of data workflow, so one of the things we immediately set up is also to have a very clear separation between directly identifiable information. This is information coded within the clinical domains on the left part of the slide here. And then we have a pseudonymization layer, which really takes care of making sure that all the data is not directly identifiable anymore to individual patients' names and birth dates and where people are living. So we have a bit more freedom in research. Still keeping in mind that we still have to comply with all kinds of ethics and regulations. But it does allow us to give a bit more freedom. And we also don't have to be that much, we don't get to see the patient names, for instance, as a researcher, which I don't want to as a researcher. And then in the research domain, we basically can add other kinds of research data, so we enrich the data with other systems. For instance, the genomics data, so we add the whole exome, whole genome, RNA-seq data. And then we have the data integration layer and different visualization tools that people can then use to actually explore and interact with the data. And in terms of the data integration platform that we have set up, we're basically using the glowing bear interface on top of the Transmart platforms. I'm not sure if people are familiar with the Transmart platforms. This is one of the systems which is actually part of the I2B2 Transmart Foundation, where people are developing or have developed actually a data warehouse that is able to code data in a structured way and make it accessible and queryable for researchers to use throughout the institute. And this basically is a user interface that allows us to select patient cohorts, and development has been going on for this interface for quite a long time. So actually, Leiden is also involved here before, but also the Dutch Twin Registry and HealthRI, which is like a national platform where they're developing all kinds of health-related research infrastructure, toolings and services. And then the data selection basically is a pretty straightforward process where basically we select all the subjects of interest or all the patients of interest that have certain characteristics. I've just highlighted a few here where I basically coded everything related to the date of birth, which is like the age at first diagnosis. You can also add which tumor type you're interested in, and then based on this selection, you get a certain number of patients selected. And then for those patients, you can then retrieve certain kind of variables or kind of entities that you want to be able to explore or interact with for your downstream tooling. And then you can export that data or link that data to your kind of favorite tool downstream. One of the features that we developed together with the Hive is a subscription feature. So this basically allows us, because some of the cancer types are actually pretty rare. In general, childhood cancer is a rare disease compared to adult cancers. And it would really allow us, it would benefit researchers a lot if we automatically get a notification if new patients come in that fit your criteria that you selected for. So this subscription feature basically allows you to save a query, and then based on that query, get notifications if new data is added or new patients are added to your cohort. And you basically get a daily or weekly update, depending on how frequently you want to get updated because of this. So switching gears a little bit, I will now talk more about kind of the genomics part. So what kind of tooling do we set up in order to get this going? And one of the things that we really decided from the get-go, which I think is also different than what happened in a lot of other institutes, is I really wanted to make sure that we have a shared infrastructure between research and diagnostics to really facilitate a fast turnaround cycle between research and healthcare. And this also is quite logical if you look at the tasks that you perform within genomic analyses. And I don't want you to focus on the specific tooling. I mean, these can be different for different kind of sets, but you can see if you look at the diagnostic settings, where basically we're using whole exome and RNA-seq as part of our standard diagnostics. And on the right-hand side, we have the whole genome sequencing analysis. You can see a lot of these tasks are actually shared. So it would really help if we have one single platform that we can actually use on both ends. That would also facilitate this kind of turnover cycle. And for this, we actually are using an open source system called Molgenis. So in the middle part, we have the LIM system. So we call it TreeCode, which is our specific instance of this open source tooling. And that really is kind of the bread and butter of the whole system. So that really interacts, with the backend. There we're using WDL and to interact with the kind of whole compute workflow, and Cromwell as a kind of execution engine. We also are using Petagene as a way to compress our data while still having it available as BAM files. So this has really helped us a lot in terms of making the data smaller but still accessible for all kinds of tooling. And at the top end, if you go towards the end users, the system will provide you with kind of morphing the data into a way that people actually understand the data. So either exporting it to C by portal, which we can then use to visualize the data for clinical end users, or use more tooling like R or Transmart itself for researchers who want to access the data either through the web interface or through the kind of more REST API kind of setup. And by integrating both the metadata with the digital workflows, and I think this is something that I'm very happy about, we can actually automate the kind of whole genome analysis quite a bit. So what we basically do is we have a biomaterial, or the material that we basically want to start with. Then we have a certain strategy, so either whole genome or exome. We have a certain analysis type, so germline, somatic calling, then single nucleotides or structure variations or copy number variations that we can call depending on the computational pipeline. We have a reference genome, so we're using HD38 in our setup, and then we have a certain workflow that we execute. And this all are basically different parameters that we feed into the system, and then basically by using the tree code system, which actually takes care of noting where the data is stored, which analysis are run at which certain time point, what data is fed into the system. And then Widow and Cromwell will basically take care of the execution on the compute backend. And at the moment, we're still running this on our local HPC cluster, so high performance compute cluster, but theoretically also given the development that has been going on with whole Cromwell interfacing, this can also be run in a cloud instance. So we haven't gotten this up and running yet, but this is something that we're looking into for both scale out as also scale up purposes in the future to be able to run it either on Google, Amazon, or Microsoft Azure, depending on which company will get a contract or get a deal with. Then just so these are kind of like quick glimpses at the kind of different solutions that we put into place. And a lot of these things we're trying to follow kind of the standards and developments. And this, of course, it's also sometimes a little bit challenging because the field is moving quite fast and also sometimes moves maybe a little bit different than you would expect 3 years ago. So some of these things really we decided 3 years ago. But what I'm really proud to say is that we actually can use this also in daily clinical practice. So just to give you one short example, so one of the things we've done for diagnostic purposes, we really have developed RNA-seq-based analysis pipeline. So one of the goals there is to really have both whole exome as well as RNA-seq part of the diagnostic process within the clinical setting. And what we've done basically is first have a number of gold standard samples, which we know for sure they're running fine with classical kind of techniques, and validate them with RNA-seq analysis. And now we've been running a consecutive study, which up until now has 257 patients involved, which we've been able to perform RNA-seq analysis on and see how well are we doing in terms of gene fusion detection. And just to give you an idea, I mean, we have the hematopoietic, so the blood cancers, the leukemia, solid tumors, and brain tumors. Well, this is kind of like a separation which I think is not that normal or natural given the occurrence of these different tumor types. If we then look at how many do we detect, so for the kind of hematopoietic or the leukemia samples, there's a higher percentage of cancers which have a gene fusion positive event within their cancer types. And you can see that this difference, of course, is different for the solid tumors as well as the brain tumors. What's really striking, and this is something that we're really happy with, is that if you compare this to the kind of standard assays, like PCR-based or targeted sequencing, and like FISH-based assays, then we basically are able to, well, we're sure we don't miss any events, so we're very sensitive, right? So we don't fail to detect these kind of gene fusion events. But what's even better is that actually we find 50% more. So we find more gene fusion events using this kind of setup. So by really using this kind of state-of-the-art genetic or sequencing-based technologies and data analysis, we can really push forward the kind of clinical setting. And this is something that, well, we're currently writing up, and we'll hopefully have really running as a kind of standard care within a few months from now. So just to give you a couple of lessons learned, so also maybe as a starting point, maybe also for the discussion later on, or the questions. So what we did from the beginning is really start and define like a data model, right? So what is now the core data model that we want to use? It's also based on kind of like the Miami standard, so this is kind of like old-fashioned maybe nowadays, but in the 90s we had the first kind of microarray standards being defined as a kind of like a standard vocabulary of how to deal with data interoperability. And also have a very clear use case, right? So the biobank is something that really touches upon all the different systems which are really needed to work in order to get everything out there and really be able to use it properly for research purposes. It's also very beneficial to share infrastructure between research and diagnostics. I mean, at the same time, and I think this is maybe also something worthwhile discuss in terms of ethics or privacy issues, it also gives you some challenges in how do you deal with the data then, right? Because some of them are generated as part of healthcare, some of them are generated as part of research, so we should really make sure that we really distinguish very well about the different usage and that we make a clear distinguishing factor there. But all of the whole genome analysis are run in research, and we're going to report certain things back to diagnostics from this research setting. So this is something that I think is worthwhile to discuss or think about. And using this automated workflow and data warehouse combination really allows us to, well, save and make visible what kind of analysis were run and what kind of results came out of those, so that in the future we can also rerun those analysis and also make sure that we have reproducible results there. And then there's two things which are kind of not related to the infrastructure, but I think very important as a kind of like a people aspect. So it's really crucial to get commitment from both researchers as well as clinicians. If you don't get that, then I think it's a lost cause. I think this is also something that we should be really aware of, that we should not only think about research, but also really think about how do we integrate and interact with clinicians, and maybe looking forward a little bit further also with patients, right? How do we engage patients into this whole discussion, which has also helped us a lot in getting high inclusion rates into people participating in the biobank study. Well, I guess the infrastructure I've already depicted here, but for us, the logistics has also been quite challenging in the beginning, right? To get everything straight and reach all the patients at the appropriate timing, especially for the ones which we only have a few days before they, from entering into the hospital to getting the informed consent signed and actually get the samples from the operation room, it can be quite challenging. So it's also something to, I think, maybe think about, debate about how to organize this. And with the last slide, I basically want to thank all the people involved. So within my group, Henry and Jane are the two team leads of the biobank and the translational bioinformatics team, and the people in like Ellen and Chris and Ryan are part of these teams. We have a very close collaboration with the diagnostic lab, otherwise it will never happen, headed by Bastian Tops, and people involved, in particular in this case, Eugene Douwe, Mark and Marco. And we also have a very close collaboration with the Holstein group, which is really also focusing a lot on single-cell sequencing, but also thinking about the genomics program and developing that as well. And then there's a number of people from the IT department, also the high-performance compute facility, and also the data stewards and the trial and data center, which are really heavily involved in discussions and making sure that we have the right clinical data, as well as our collaboration with the Hive, which has really been fundamental to develop the whole Transmort Glowing Bear interface. And with that, I would like to thank you for your attention. Do you want me to plug it back in again? Or,
yeah, sure. Okay, thank you, Patrick. This was great. So our last speaker is still going to be online. So Sumit Jamuar is from the Global Gene Corp company. He had actually an interesting trajectory coming from finance and consulting into our world, and he's going to talk about bringing diversity into genomic and health.
Thank you, Moran. Can you hear me? Just checking if you guys can hear me. If so, please raise your hand. Andy? Yep, great. Now, I will need help in terms of slide transition as well. If someone may be able to do it on that end. So from my perspective, look, I could not match up with Patrick and Barend in terms of the technical content. So what I wanted to bring in today was more on the layperson's perspective on why I'm personally so excited about genomics and the transformation that it's bringing to the world. From my perspective, first of all, apologies, I couldn't make it. I really wanted to be there because last time I was at the GA4GH meeting, it was absolutely phenomenal. And you met some old friends and made some new ones. In lieu of me, I've got someone who's much better than me out there in the audience, and that's our co-founder and co-chief scientific officer, Dr. Jonathan Picker, who's based out there, and he's there, and he'll be happy to take some questions offline, after this session ends or during the interaction. Andy, just wanted to check: is the presentation working, or should I try to?
We're still just trying to figure out how to actually bring it up at the moment. So.
Okay. Well, why don't I try to see if I can log into my because I've got the presentation right here. So let me just see if the Zoom works. Then we are all sorted.
Might be able to do it off.
So there are two views of me coming on. Is this working now?
That is working, yes.
Oh, brilliant. Okay.
Fantastic.
So I, from my perspective, I think the bit which I'm really excited by is if you think about what technology has done over the past few decades, and the impact comes back to what it means for each one of us. I mean, this graph is a startling graph where when I look at the world as well as in terms of average life expectancy at birth, and look at India, the average life expectancy in 1947, which was the time of Indian independence, was about 32 years. And now it's about 69 years, really close to where the global average is. And much of it has been driven by all the advances which happened in public health and the transformation that took place due to the work that went on there. When we look at the next big challenge is all about genomics and what does genomics mean for each one of us. And I think what excites me personally and our team is that genomics is driving the realization of human potential, both in terms of quality as well as quantity. So the question we always ponder when we think about the team is to say, look, if you had a meta objective, what can we do in terms of making 70 or the health that we have at 70? What can we do to make 70 the new 40, right? How can we extend the healthy lifespan to as long as possible? And, almost doubling that healthy lifespan is an interesting target. And then what that drives is a variety of other benefits in terms of longevity and other things. But the core is, can genomics allow us to understand what it means for each one of us in terms of what it takes to be healthy and how we are going to, how and what we need to do in terms of making that happen. And the two trends we are taking place, I mean, this room is phenomenally familiar with this curve, so I'm not going to get really into detail. But what I look at it as an outsider coming in is, first of all, that
we've just lost you for a second, sorry.
this curve is good.
Sorry, we just lost you for a second there, for about 30 seconds or so, sorry.
Oh, can you hear me now?
Yes, we can.
Great. So the question is around, when we look at big disruptions in terms of technology changes, everything comes back to what is it, not only that is the technology good enough, but are we able to do something with that technology? The second thing, which is really exciting, which again is, something that people in this room are driving and building on, is the fact that you have AI and machine learning, which is much better than what it was, over the past decades, and it's just going to improve. So this is a very famous chart which floats around, which is that AI has been about 1 million times better than what it was in the last 30 years, and it's going to be another 1 million times better over the next 30 years. So what we have is the ability to not only sequence it, which is convert the biological information into machine-readable information, but it's also about converting it into being able to analyze and interpret and find the patterns onto it. And if you think about what's happened from a genomics perspective, the first big revolution was all about sequencing, from the Human Genome Project to the machines which are available now, which allow us to do the sequencing. The second was the problem that the industry and researchers got together to solve was the analytical tools and the bioinformatics tools which are needed to make that, to be able to analyze the data. The third big change which has started to happen is, I think, is twofold. The first is around application, because we've been able to apply it across different industries. And so if you think about the whole promise around healthcare, it's from treatment to wellness, right? It's about if you fall ill, what to the right person, what's the right drug, what's the right dosage, what's the right time, the whole concept of precision medicine. But it's also almost saying that, hey, even before we get to someone falling sick, can we prevent that sickness? Because that will be something which is truly, truly exciting. And one of the disruptions in the use cases that's happening is all around a pharma R&D example, right? Where we have seen that if you think about success rate, without genomics, it's 8% of all molecules which enter clinical trial 1 make it to the other side. So that means just over 90% failure. Time to market is 10 to 15 years, which is from start to building and getting it out there. If you succeed, it's 10 to 15 years with all the data and things that are associated. And the third is around cost per drug is about $2 billion. Now, when you add genomics and biomarkers, already there are indications that you have a 3x improvement in success rate. Time to market goes down by almost 4 times, and you have a cost of, cost of per drug goes down to half of it. And again, the last time we saw such a fundamental shift driven by technology was when I was growing up in the internet revolution, which is the 90s, when you studied and you saw these excitement around a technological disruption with different business model innovation that is transforming the way different industries were run and disrupting really big pools of spend. And that is what genomics is driving. So if you take just the pharma R&D market, that's about $200 billion annual spend. So that's one element which is coming into play. The second is around data. And that's where one of the biggest challenges that we have in genomics is that, this is a paper by Sarah Tishkoff, which was published in March 2019 in Cell. And still almost 80% of all data is from people of European ancestry. I looked at the national initiatives breakout earlier. All of those initiatives are linked to Canada, Australia, and those are still essentially solving and cataloging a really important aspect, but most of it is around the European ancestry. When you look at a place like India, for example, India is about 25% of Indian subcontinent is about 25% of world's population. It contributes less than 2% of genomic data and insight. And the reason why this matters is, if you go back to data sciences, we have the tools, but if you don't have an unbiased training set, we can never achieve the right outcome, right? We cannot get to the right answer. And if you add the other complexity around the number of sort of subpopulations and ethnicities that exist in different countries, the Indian subcontinent has about 4,500 ethnicities, which is almost 50% of the world's ethnicities and subpopulations. So we have a really rich source. And if you extend that to Africa, to Middle East, and Asia, rest of Asia and Latin America, there are really rich sources of training sets which are there, which are what are basically causing us to not achieve the full promise of genomics, all the wonderful stuff that we talk about. So we've had to deal with everything that Barend and Patrick talked about, which is, people say, why does it matter, right? And it matters because some of the early work that David Reich has done was around the PC analysis around different populations, showing that different populations are quite, there's a certain distance around them and they have different characteristics. And that is something that, if you go to what is the known knowledge about different populations, there is a lot around, each of these clusters. Hi, can you hear me?
Yep, you're coming back now.
So each of these clusters and different colors clusters represent a different sort of population, as to say. And if you look at the pink blob, which is almost 50% of the richness in terms of ethnicities and the characteristics and other things, you have, that is characterized as one big blob, which, as a data scientist or as someone who's looking at finding answers, it's about, what's the right level of resolution that we need. So we started about, as Moran rightly said, look, the thing which intrigued me was I came from a different background. I'm a chemical engineer. And just over 7 years ago, I met with Jonathan in his offices in Boston, in BCH, and Soumya. And we started discussing what it meant. And I was just smitten about this idea about genomics. And I said, this is incredible, guys. Can you do this for me? And they said, no, we can't. And I asked them, why not? And they said, it's because we do not know what a good Sumit looks like. So I can sequence you, but I can't give you anything relevant. So we had to then, as we tested some of the known knowledge about different populations, particularly in South Asia, we found that some of the insights and understanding when we looked at case and a control set were inaccurate because they did not correlate with the phenotype or the observed phenotype behavior. And the genetic data did not match up to, or the genetic insight or the known insight did not match up to it. So what we have then been doing is we have been mapping and organizing the diversity. And it touches upon every aspect of it, where it starts off with consent, something that, GA4GH and our colleagues in GA4GH are defining, how we think about ethics around this. But it's about taking and creating the right consented framework with the right IRB approved stuff. The second thing is, how do we create clean sources of phenotype and clinical data, right? So we do deep phenotyping in places where no EHR exists. And in some ways, it's a plus and a minus. The minus is EHRs don't exist. The plus is when you're creating a training set, you can actually start from scratch and use standardized methodology, ontologies. We created technology-driven phenotypic execution tools, which means that when our bioinformaticians or our partner bioinformatician or you want to look at a particular thing, all you need to do is pick up the code rather than spend the 60% that Barend talked about in terms of cleaning up the data, right? So we can get straight on with doing the experiments and testing our hypothesis rather than waiting for the data to be cleaned up and still having that. The third aspect is the other aspect, which I think Patrick mentioned, which is the hard bit about, transporting biosamples. So we take blood. How do we transport it within a certain time period, within 24 hours, from any point in India or anywhere else into our CAP certified lab? How do we do it in a way that it allows us to, extract the DNA at the right quality to be able to? So it's a hard problem because it's the right ethical framework, along with the right phenotypic and clinical characteristics, which then, because we have the consent to capture it longitudinally, so not only do we do a deep snapshot, we have the ability to capture it. And the third is dealing with biological material with the right, again, which has to be transported in a certain way. So as we do that, what is really interesting is that we started seeing really high resolution aspects within the populations. And I'll just touch on the ancestry side because that seems to be a theme people are looking at. So, we just took 10 out of the 4,500 ethnicities out in India out of scientific curiosity to say, look, are they all similar? In which case, do we need to look at everything? Or do we just look at it as do they cluster on some things? And when we did the PC analysis, what was interesting was that even these ethnicities or subpopulations started clustering around and had very clear patterns, right? And that was quite an interesting thematic. And from then, what we did was we worked on saying, okay, if that is the case, can we do some, statistical analysis to look at components? And this is just for 3 states, there are certain subpopulations. And what we're looking at is each vertical line is looking at what the composition is of that particular ethnicity versus the other thing. So it's trying to look at the admixture events. And there is very interesting and very high resolution insights that we are starting to generate around that. And, and that richness is really critical because if we want to truly, we've got the tools, we've got the use cases, and then new use cases being created. If we can really do this appropriately, that really structured data is what each one of us in this room needs to be able to sometimes run our experiments to get, decipher the signal versus the noise that exists. And on that note, I'm just going to wrap up on this and saying, look, it's really hard work, right? It's not something that one person, one organization, one entity can do. The people sitting in this room are the people who have the right motivation, who are aligned with our values and visions of looking at this and saying, what can we do together to solve this problem, to make sure that the benefits of technology appear to every person and are delivered to every person in the world. It's not just a few people. And even for those few people, there's a limit to what genomics is able to do because we are unable to look at certain diseases in the way because we don't have enough data, so we can't solve for disease. So what can we do in terms of working together to map and organize the world's genomic diversity and make it useful and accessible to deliver high quality life and longevity for each one of us? And it's not just about an esoteric pursuit of certain research, but it's about converting that esoteric pursuit and the insights which are driven by folks in this community and the GA4GH community to then deliver it to the 7.5+ billion people out there. And what excites me is, when we started 7 years ago, most people said, you're crazy. They said, why would anyone care about diversity? Right now, the biggest topic about genomics and what's holding us back is the fact that we do not have high fidelity data, which is representative of the richness of our world. And the second criticism or comment which used to come our way was, look, who's going to pay for this? Why would anyone, people can't afford to pay thousands of dollars to get the genomics inside. And I always draw back to a technology which we have seen, which is the mobile phone. I mean, I don't know how many of you remember the mobile phone from 1980s. Has anyone seen it? Show of hands in the room. How many people? Yep, so one. How many people remember the iconic 5510 Nokia? Most of us, I used to call it the brick because for weeks on end, the battery did not run out. I could throw it to do anything, but it still survived. A friend of mine even tried running as geeks do. They ran their car over it and it still survived and it was still working. So that was how solid that was. And then the BlackBerry to the smartphone to everything else that was happening. And when you think about that revolution, when mobile phones first started, they said, it's for the folks in Wall Street, the rich guys who are doing it. Right now, the mobile penetration in Africa, India, China, Latin America is significant. India has more mobile internet users than US, right? And that is the power of transformation through technology. It changes behavior. And what I want us to do is imagine how that behavioral change will happen. And imagine if together and what we are creating, if we can turbocharge our mission by creating the data operating system so that we can just get on with our hypothesis, get on with our experiments to have an unbiased training set. And so that is the mission that I just wanted to bring to your attention. So if I were to summarize, I mean, look, this is where we are. We are based at the Welcome Genome Campus and Boston and Singapore and India. So you're more than welcome to join us at any point that you want. But I'd just like to summarize in saying, like every great technology, this technology revolution is going to impact each one of us and it is impacting each one of us because it is fundamentally changing how we interact with ourselves because the data and then the use cases are making a difference. That is life-changing in certain cases.
Sumit, thank you very much.
The changes that happened. The first was all about sequencing. The second was all about analytical tools. The third is all about the data and the used applications and disrupting industries across the piece. And what we need is, and what we're building is creating that unbiased data set so that we can get on with creating more use cases and delivering more value for every person on the planet. And
Sumit.
so I would just pause and stop there. And again, thank GA4GH, you, Andy, Moran, and Justina for inviting me to this conversation. And thank you so much for your attention.
Thank you. And thank you, everyone, for sticking through our technology challenges and so on. So perhaps we'll take a few questions from the audience. So I invite you folks to, actually, we kind of set up a little stage over there, if you'd like to sit. And I wonder if we have any question from the audience? No? Okay. Oh, great. Wonderful. Jonathan, thank you.
So are there any questions?
Okay. Maybe we can start.
I'm
Andy.
quite happy starting off with
Yes.
one. So some of the points that were raised here were about hybrid cloud solutions as well, a way of actually combining local compute with cloud compute. And what I'm wondering is, what do the panel feel are the challenges surrounding that environment when we start to talk about personal data and patient data as well? Patrick might be the best person to start that, I think, but...
Is this on? Okay, I think so. Well, there's certainly a couple of challenges that we have to deal with there. And at the same time, the way we're thinking about it now is basically, so we have our local compute facility where we can run most of our analysis on, and we're thinking about putting the data into our cloud system to make it accessible also for other research outside of the institute. And to have a platform where basically we can put all the data on, and then we can give access to the researchers outside of our own institute, which can then use that data and also do the compute on there. And I think it's moving forward in the sense that it's becoming more common once you have the right contracts in place. So I think this is something that also the whole GDPR discussion in Europe, and I guess also here in the States, has been really helpful, that there's more and more attention are drawn towards having the appropriate contracts in place with companies like Microsoft or Google or Amazon, and have them part of this kind of ecosystem in this setup.
Yes, I think actually this is perhaps one of the critically important questions that we're dealing with, not just hospital data, but general population. Certainly, as we look around the world, there are countries and people who have experienced in the past of data and information and resources being used from the West without them having control of it. I do think that Barend Mons described it very nicely. The provision of metadata is one secure way to go, and then allowing researchers who have the due consents to do IRBs to come in for more of the data access that would be relevant is perhaps the way to go. And again, those IRBs probably being generated locally to ensure the buy-in from the populations who are being interrogated.
Yeah. Not sure if I have many cents to add to this. What I think happens. So I'm not a cloud technology expert. Just put it out there. But what I think we are sort of achieving with FAIR principles, also with the standardization work going on here in the Global Alliance, is I guess we're sort of decoupling this. So the cloud computer becomes an implementation issue, and it's much more about, my colleague here said, about by standardizing, by contracts, actually put the right people in control over what they need to be in control over. Then the cloud computing, whether it's local or whether it's somewhere else, but with all those contracts, it becomes an implementation issue, not so much a forefront issue for users.
There's an ethical discussion which I think is linked to also what you said. It is like, to what degree do you want to share the data, and to what degree do we inform also the patient or the subject who actually gave the data to a certain institute, right? And this, I think, is something that's really, getting more attention now, and also involves a lot of patient advocates and organizations really involved in this. And it's also maybe sometimes a personal decision, or actually it is a personal decision. But to what degree do you trust the organization where you put your data at, or do you want to be asked every single time someone is using your data for a given purpose, right? So this is something I think and be part of it.
And in my experience, I work a lot in the rare disease community, and we have some of them in my family, so sometimes on the other side. And what I find really interesting is that humans are so incredibly inconsistent. So if you would ask patients, especially policymakers, it's much about control. At the same time, the same patients put information on Facebook to share with their other patients, which is really useful for other patients. So that's socially very interesting.
I think one interesting aspect that was mentioned in the conversations in the context of FAIR is making things accessible for AI. But the key thing is, if you want to join data together, and I think this is what you folks are talking about, is the selection of ontologies and selection of models such that actually all the data sets will be interpretable together. Can you comment on when you were implementing your systems, for example, in the Princess Maxima Center, how did you go about selecting the model? How did you go about selecting which ontologies? There's like so many options, and at the end of the day, it's so detrimental with regards to how your data set will be able to be combined with others.
Well, in the end, I mean, so the way we started basically is to start with a common number of like general ones which are quite widely accepted. But you're right, I mean, there's still options to choose from. So we started basically actually with the sequencing, like the SRA EGA model, which was really helpful in getting lots of the things done. And then also looking at kind of like standards for how do you describe your study and your cohort and those kind of things. What I think, so that's one thing, right? Selecting the right ontology. But the other thing which we also encounter if we want to compare data, I mean, even though, for instance, in cancer, but also in pediatric cancer, the ICDO is one coding system that we all use throughout the world. I think most cancer institutes use those. But depending on the level, you annotate it, the samples, you can actually not compare it directly, right? So for instance, we now have a case where one institute says, okay, it's medulloblastoma, but actually we know for medulloblastoma, it's actually there's a WNT SSH group 3-4, right? So subtype. So if you don't have it at the appropriate level annotated, then you even have the same ontology and use the same terms, but still you can't compare the data directly. So I think this is also something that we should talk about. How do you organize this globally?
You're absolutely right. And one of the concerns we had was you may take one or two ontologies and then use them to extrapolate to the other ones because people have cross-translated. One of the issues we've dealt with is you have to go even further back, right to the root phenotype in many cases, particularly when you're working with populations that have very different language, not just in terms of the language, but their language for describing symptoms and signs. I remember I trained in the north of England where I learned that bladder warts actually meant bladder cancer. And in fact, the way we've taken the approach is we take these terms, we find out what they actually mean, and then translate them into each ontology independent of the others to try to avoid a Chinese whisper situation of increasingly becoming less accurate.
So maybe... So I work especially in the rare disease domain, but in general, actually, we work on implementation of FAIR, the verification workflow, and this. I think it's also good to be honest about this. This is to a large extent an unsolved problem. We kind of also created the problem. First, we had 10 years ago, no ontologies, and now we have many for the same thing. So we created that problem. I hope that in a couple of years' time, we'll have better answers. There's one thing that we often emphasize is that at the moment we are emphasizing the role of data stewards, who are the sort of hybrid people between technical experts, ontology experts, and people really working on the domain, working on cancer. Because I guess together and with these people as sort of central persons, we need to resolve this issue for now, and hopefully over years' time, be able to give better answers to this techie question.
I think there's a question from the audience.
I hope the mic's on.
You're welcome to come here.
Okay. All right.
I think it is on, actually. I just try that one.
Hi. Can you hear me?
Yes.
Okay. Yeah. My name is Mohamed Simatsaba from Botswana, and I'm representing H3 Africa. So I have a couple of questions. I think one, I'll start with you, Jonathan, since Sumit is not here. In your engagement with the community, specifically in India, because I think there will be a lot of similarities in what we are dealing with in Botswana, or in Africa in general. Were there any issues in terms of fear of discrimination and all of this as you introduce this new technology? There's a lot to learn, but there's also a lot to benefit. But what we are dealing with in Africa is where people are fearful as to people using this information, especially large-scale genomics, like we are discussing? And if so, how did you engage the community to make sure that you maximize out of that? What we look at in H3 Africa is generally capacity building, because as a continent we are behind in terms of technology, human capital, and all these other things. Are you also imparting all of those in India as well? And lastly, think about collaboration, and I'll be happy to talk about to you about that. The next one is to Patrick. A great work that you're doing with genomics and cancer in children. We are actually in the process of setting up, not exactly similar, we don't have the amount of money that you have, but we aspire to have a very, very similar system and outcomes and all of that, where we are setting up clinical services and at the same time thinking we should be setting up on the research side, biobanking and almost what you have already done. So is there any advice in terms of what should we not focus on? Right now we are looking at which equipment to buy and which systems to put in, which EMR and all these other things. Is there anything that you look back and say, we shouldn't have spent money on that? I know I'm putting you on the spot, but we are also trying to save money at the same time, but get the same outcomes. Your second question would be, we have issues with pathology and all these other things, and that's another problem on its own. Have you used gene panels or whatever you use in reverse now to diagnose rather than for prognosis or treatment and all this other? Are there examples that we can learn from? Because I think we can try and leapfrog, as in the last example, go from the big telephone to the smartphone without going through the rest of the process. Thank you.
Thank you.
Well, thank you. That was actually a very interesting question. It was one of the very early ones that we addressed, was what was going to be to partner with populations in India in order to obtain both phenotype and the material for genetic analysis. Now, interestingly, I will say, as we've looked at different populations and different countries around the world, not surprisingly, there is no generalization. Different people look at this very differently. What we do share in common is the infrastructure needed to reach populations that may not have the same resources can be shared, but the actual process of getting them to engage is very differently. We carried out a survey fairly early on in India to understand what the people would want to be involved. To our surprise, overwhelmingly, what they wanted was to know that the data was going to be used to improve medicine for their community, their families, and it was at the end of the day altruism that was driving them. This has not necessarily been the case in other populations. What we've done as we've spoken with each is we feel that we need to engage with the community and work with them in partnership, and that's really what makes the difference to be able to go forward.
Okay, just to respond to your two questions. So it's maybe also good to talk offline afterwards, because I've talked to Nicola in the past about H3 Africa, but we should talk offline. So regarding to your question about what you should invest in hindsight, so I think, I mean, what we've also learned during, while setting up the system, actually, so I've always been an open source, open data kind of fanatic, and really trying to push that forward, and I think that has helped us a lot. And the systems where we actually have most difficulty with getting going are really the closed commercial solutions that we are faced with, you know, from the kind of more clinical perspective. It's very hard to get them to move more quickly in terms of this kind of things we want. So whenever you can choose for, you know, having at least your data model open and having no discussion about how does the data model look like, that I think is something really fundamental to look into and really make sure that you have access to the data. Not per se that the solution has to be open source, but the data model that underlies the whole system has to be open and accessible for interpretation. Otherwise, you can never, you know, have thorough debates about that. In terms of the gene panel, so what we do, we basically have now a virtual gene panel. So we do whole exome analysis in research, and what we report back to diagnostics is about a list, and I'm looking at Jane, I think it's about 400, right, that we look at. Yeah, so we have about 400 genes that we report back to diagnostics about, and that's what we tell them, basically. We don't tell them about the other things. And that's kind of the agreement we made them. So you could also design a panel for those 400, right? You go back to kind of like cheaper assays, like a SNP or assay where you actually use those kind of panels. I mean, we decided not to do it because, like you said, it is a money issue, so that will cost you more. But the advantage of using the whole genome sequencing data or exome sequencing data is that we can easily switch in the future also to a different gene panel or add certain variants to it because we already are doing a whole exome. So that kind of benefit you would lose. But of course, a SNP array is much cheaper than doing a whole exome. So this might be a cost aspect you want to take on board then as well.
And maybe there's one thing I can pitch because it's about Africa. So I'm speaking for the FAIR part. So there is a global open FAIR implementation network specifically for Africa. That's being I think it's a starting network, and so you'd have to look for Google for Go FAIR Africa. And I think that's interesting. It's one of the networks. I'm doing one for rare diseases about how to change culture, which is very much needed for FAIR. Then this one is addressing how does that apply in the context of Africa. So that might be something to look at.
Thank you very much. I want to thank everyone for your time, for your answers, for your presentations. Thank you for participating.